good afternoon. This is Pam Todd. I'm Pam Todd, and I'm on the patient engagement team at Global Genes, and I was deep into data DIY last year, so I'm really excited about this further iteration. We're going to be talking today about translational research from academia to industry. So I just wanted to give you a few little housekeeping things, which I'm guessing you've heard before, but just to reiterate, if you have questions, you are muted. So we ask you to put them in the chat, and just give us lots of questions. Just everything you've ever wanted to know about academia to industry, please put it in the chat box, and I will be passing those questions on to our speakers at the end. We'll have about 15 minutes at the end for questions. If we can't get to your questions, we will still be gathering those and asking our speakers if they'd be willing to follow up with responses, and we'll get those answers to you. Again, if you have any technical difficulties, just go back to the lobby, click into the help desk, and you will find a whole team of people to help you there. They've been keeping us on the, you don't see it, but what looks like magic is a whole team behind the scenes making sure that technology is always working, which is great. So I would just like to introduce our speakers. By introduce, I mean I'm just going to tell you their names and their affiliations, and then they're going to follow up with their a little more information about their backgrounds as they present. So first we're going to have Tamar Grossman from Arcturus Therapeutic. I hope I'm saying that right.
Yes.
Thank you. And Ethan Perstein from Perlara. Tamar's going to begin, and then she'll hand it on over to Ethan. So welcome.
Thank you so much, Pam. And hello, everybody. I can't see you, but I know you're there. I'm Tamar Grossman. My background is all rare diseases, genetic diseases. I've done my PhD at Tel Aviv University and my postdoc at UCSD, working on various mechanisms and animal models for rare diseases, including Rett syndrome and epilepsy modeling. And about 10 years ago, I moved to industry to work at Ionis Pharmaceuticals, where I spent the past 9.5 years working on taking the various rare diseases from the research stage, and some of them all the way to development and clinical trial. And a lot of those were initiated by actually academic lab and patient organization. And I'm going to share my experience today and talk about one disease that I chose to talk about, which was a collaboration with academia, and this is a drug that will be in patients soon. A couple of months ago, I transitioned to work for Arcturus Pharmaceutical as the head of rare diseases, working on various rare diseases therapeutics, still in the RNA therapy space. And I'll be talking also about my work with the Cystic Fibrosis program that we have that is also shared in partnership with the CF Foundation. So let me share my screen and we'll get started. Can you see it? Okay. All right. So I'm going to talk to you today about RNA therapy for rare diseases from academic research to therapy. And as you all know, there's a huge unmet need for finding therapies for rare diseases. You've probably seen these numbers multiple times. There are 7,000 different rare diseases and about 30 million Americans are suffering from rare diseases. So that makes it 1 in 10 Americans that have a rare condition. And there are only 575, and probably a little more because this is from 2017, which really covers 5% of all diseases. So there's an unmet need, and there's promise. There's a lot of drugs in development, about 560 at the time that this was put together. And they're for different disorders, but I'm sure that some of you that are listening to this know that, some of the diseases are not covered. A lot of them are not covered. And the purpose of this whole event and my presentation is to ask, what can we do and how can we put more drugs in the clinic and finding more cures and therapies for rare diseases. And how do we do it? How do we pursue rare disease treatment? There are four really key factors that determine the feasibility to do so. And they are really forming a sweet spot for drug development. So you really have to start with a validated therapeutic targets. That means that it's a gene or a protein that you know that if you target it, you get improvement in disease. You have to have a clinical data set. This means that you know the disease, the natural history, the progression, what are the biomarkers. There have to be a regulatory path to know that you can take this drug and get it approved, and an addressable patient population. That is always key for big pharma because they want to be profitable. And if there's only one or two patients, that's not going to make them the big bucks. So that is changing. And as you all know, there are all kinds of initiatives that are changing. And there are more and more drugs that are geared to ultra rare diseases and even N of 1. And I can't wait to hear Ethan's presentations next, who's going to be focusing on these aspects of this ultra rare and an N of 1 trials. So, but all of them ultimately bring the sweet spot for drug development. And before I move forward and tell you about the examples, and one of my examples is actually an ultra-rare disease, Lafora disease. There are maybe 200 patients worldwide. So I want to give you the path and just a little overview and what it takes to take a drug from the idea from the academia, with the academic lab, all the way to drug development. What are the steps? How long does it take? And what's the cost? So the start is usually by exploratory research. This is usually done in either academic lab or in early research phases where you find the target, you validate it, and you have data that shows that it works in the animal model. The next step will be to find the human drug. Then you are doing some high throughput screening. There's more resources that are put into this phase to find the drug and start working on the transition from the research to development. How do you take it to men? And the next step is the preclinical development. You start manufacturing the drug in a GMP manufacturing and start doing studies to show that the drug is safe. And these are the toxicology studies and PK studies. And at this stage, you may start doing natural history study because we've learned in the past 2 days how important these are for clinical development, especially for rare diseases. The early clinical stage is the first human dose, and in rare diseases, that may be a patient. And that's why it is important to have the natural history study, because most of those studies are done without the placebo control, meaning all patients are getting the drug, and the outcome is compared to historical control in the natural history study. From this stage, you move for late clinical development, and then you have to demonstrate a broad efficacy in multiple patients and take the results and move to regulatory. But what does it take? How long it takes? How much it costs? These are estimated number for drug discovery. And you can see that it goes from minimal resources that are putting into the research all the way to development that shows that a couple of millions. And the estimated time in years for developing a drug is about 7 to 10 years. And about $30 million. But with the rare diseases, it's lower because you have smaller trials. There's an initiative and guidelines for rare disease regulatory processes and development, and the numbers could be lower. So I'll be giving you examples today from 2 different therapeutic approaches that are all focused on RNA therapeutics. And just to remind you, if there are no scientific background, just a dogma of DNA where you are having your genetic code, and this translated to message RNA, which it translates by transcription, and this is translated to the protein, and the protein is what makes all the functions in our bodies. When we talk about the diseases, you have mutations in the DNA. You have a mutation that carries into the message RNA, having a defected protein that could aggregate or cause problems and cause a disease. So the first example I will talk about will be the ASO, which is an antisense oligonucleotide, but that is a piece of DNA that binds the message RNA and stops its translation to proteins. So basically, with diseases that you have a toxic gain of function or problems that are generated because you have a mutant protein, you eliminate that by targeting the antisense oligonucleotide. And the story that I'm going to tell you is about Lafora disease. Lafora disease is a rare and a fatal autosomal recessive disease. You need to inherit 2 copies of the mutated genes from both parents to have the disease. And it's characterized by accumulation of Lafora bodies. And Lafora bodies are basically unbranched glycogens. So glycogens accumulating in multiple tissues, but most importantly in the brain, it causes dysfunction and of the neurons, and that leads to seizures, epilepsy, myoclonus, and a very fast progressing disease leading to dementia and death within 10 years. The children start get their diagnosis usually at late childhood and adolescent at age 10 or up to 17, but they don't live past the age of 25. It's a very severe disease and there's no cure. And my experience with the Lafora disease was initiated basically in a rare disease conference when a colleague of Dr. Minassian and I had a conversation of a similar disease, which is called APBD, the similar process, and brainstorming on how can we do something for this patient? What can we do? What can we target to help the disease, to cure the disease? And we're thinking of stopping glycogen synthesis, because if you stop glycogen synthesis, you'll stop forming those Lafora bodies and stop the disease. And we've decided to target GAIS1, this is glycogen synthase, and hopefully show improvement. Dr. Minassian works closely with patients with Lafora disease worldwide and with the foundation, the Chelsea's Hope, and also individual families and patients around the world who contribute also funds and resources for his lab to do the research and finding the drug. And throughout discussions, he initiated some studies to test the hypothesis whether inhibition of GAIS1 could improve the disease. And he generated that by genetic models in the mice, and he was successful because we were able to show, he was able to show basically, and others, that when you inhibit glycogen synthase, you move from, as you can see here, seizures and disease from in the mice to basically a wild type or a normal phase. And when you look at the brains, when you eliminate that glycogen synthase from the brains of these mice that have Lafora disease, it clears their brain from the Lafora bodies. And all these dots that you see here is an accumulation of Lafora bodies and glycogen. And when you knock down this gene, even 50%, you get significant improvement and the brains are looking completely healthy. Dr. Minassian also introduced me to the key investigators and the foundation, and we all worked together and communicated throughout this process. These piece of data that he shared with me gave the rationale to move forward at Ionis and develop the mouse drug to test it in the animal model. And through high throughput screening, I was able to identify some oligos that could stop production of glycogen synthase and were safe in vivo to take forward to the model. And we sent those oligos to sick kids, and Dr. Minassian and his postdoc were testing them in the animal models. And the data was really exciting because we were able to show that with our drug that had to be administered directly into the brain, we're able to clear those brains from the Lafora bodies. And this is a quantification of the brain showing that when you have the drug administered, you have no Lafora bodies. So we had the rationale to move forward because we could show a proof of concept in the animal models showing benefit to the mice that have Lafora disease. So the next step, we started screening for the human sequence. Since these are sequence specific, we had to go through the process of finding the human gene and do safety and tolerability studies. At this point, we also initiated a natural history study. We worked closely with the foundation and with families and incorporated all their input on what's important for the patients, what are the different biomarkers that we can add, and all the potential endpoints, and all that was incorporated into the trial design that will hopefully start soon. And the idea is to dose and give the drug to all patients and compare that to the natural history. So this is an example of a program that started with a very eager, enthusiastic neurologist and myself meeting at a rare disease conference and moving this forward. Hopefully we'll get into patients very soon. The next program that I want to talk to you about is the current program that I'm working on, which is the cystic fibrosis program. And in this case, you have a mutation in the DNA that encodes a defected RNA, and there's no protein. So there's no CFTR protein, and that is what's causing the disease. So what happens with our technology and the idea is for the therapy is to deliver the normal CFTR protein. So the normal sequence, let the body make the protein through the natural processes for translation and have a healthy outcome out of that. So this is called Lunar CF, the drug, and the program is for therapy for cystic fibrosis. So what is this Lunar? These are basically a vehicle of nanoparticles that carry the messenger RNA into the cells. It has a lot of different uses for administration by intravenously, intramuscularly. We have a COVID-19 vaccination program using the same technology. And for the CF, we're using nebulization, which is basically administering the drug by aerosol. We have over 200 proprietary lipids that can target multiple different cell types. So there are many diseases that we can target by introducing the normal healthy copy of the messenger RNA. Another important point is that those particles are biodegradable. Their job is to take the messenger RNA, get it into the cell bed, and then they degrade and disappear. So you can't even trace them in your body after administration. We have multiple programs. Our OTC is a rare disease program that is currently in phase 1, and there are others. I'll be talking about CF. We have multiple collaborations with other pharmaceutical companies like Ultragenics for glycogen storage disease type 3 and other rare diseases. And I'm going to focus today on the program of cystic fibrosis. So just to give you a little background, for those of you who are not familiar with the disease, cystic fibrosis is a rare inherited disease. It affects multiple organs, most importantly the lung. And this is because CFTR gene is mutated, and that decreases the movement of chloride ion outside the cells. And that results in a very thick mucus layer that covers all airways in the lung, and that causes inflammation and infections and tissue scarring and respiratory dysfunctions. The patients have to go through multiple different treatments during the day. They sometimes spend hours getting different drugs. As you can see here, this is an aerosolized drug that the patient's been treated in. The vest that the patient is wearing is a vibrating vest to massage the lung and help reduce the mucus and movement. So it's a huge burden on the patients, and there's still a significant unmet need. There are recent drugs that were approved by Vertex, and they are doing great for some patients because there are mutation-specific drugs. So there's no actual drug that can treat patients in need. There are 70,000 patients suffering from cystic fibrosis worldwide, and there is a portion of them with the null mutations that generates no protein that cannot take any of those drugs that are in the market. There's a significant unmet need for this patient population, and our approach will be to do a replacement that will deliver the new copy and a normal copy of CFTR into the lung of the patients, and will be independent of the genotype of the disease. The functional CFTR protein, hopefully once in the cells will be clearing the chloride and will reduce the mucus, reduce the scarring, and hopefully improve the progressive respiratory dysfunction in the patient. So how it is done, we generated the normal message RNA. This is the cargo. It goes into the lunar particle, which is aerosolized into the lung. In the lung, we've shown that it can distribute to lower and upper airways, and this will express CFTR and hopefully restore normal function. We've shown that with the lunar formulation, we're able to deliver it specifically to the lung. These are mice that were dosed with the lunar carrying a luciferase. This is a way for us to monitor where the drug is distributed, and you can see the nasal area where the drug was administered and the lung. And when you section lungs that are treated with another marker here, you can see that these lunar formulation can deliver the cargo to the relevant cells in the lung, which are the branchial epithelial cells. We've done these kind of studies in multiple species, in rats and mice, ferret, and also in monkeys and non-human primates. And with all of them, we were able to show that we're getting a specific distribution in the cells that are relevant for disease. These are the epithelial cells here in brown. And in a higher magnification, you can see the different cell types and the nice specific distribution. Most importantly, we were able to show that we can restore function of CFTR. We used the nasal potential difference assay. This is an assay that's also used in patients. And in the patients, and in the mice who are patients in this study, we administer the lunar CFTR by intranasal administration. This gives us a snapshot of the lung, a non-invasive way to really get efficacy and see if the drug works, because the same epithelial cells that line up the nasal epithelium are very similar to the lung epithelial cells. And we were able to show that when we administer the drug, we not only get it into the right cells, but we get activity. So these are channels, and by movement of the chloride ions, you can generate electronic current that you can measure. And this shows that with the treated groups, we get significant improvement in the expression. And these are CFTR knockout mice. So they are like patients that carry the null mutation. They have no CFTR activity. So they are basically at the zero current, but when you administer our drug, you restore the function of the CFTR and get activity. And this is just done with different formulation that we tested, showing consistently that we can improve and increase activity of CFTR. We've done many other studies that I can't share with you, but I'll just give you some highlights. We showed that these is a really optimal vehicle way that we can aerosolize. And we've shown that the lunar formulation of the nanoparticles shield and protect the message RNA in CF mucus. It is, we've shown that with double stains that we are targeting the correct cells in the lung, which are the ciliated cells that will contribute to the improvement. And we've optimized the sequence of the message RNA to ensure that we're having high protein level and biologically active protein. This whole program was supported by the CF Foundation. As you know, the CF Foundation has been very active and working closely with academia and industry to promote drug development and discovery for cystic fibrosis. And they actually led to the discovery and the approval of the therapies generated by Vertex. And it's been a really fantastic experience working with them and pushing this program forward. But to close this, I would just gonna go back to what we started with and how important are the activities and the contribution from the patient-led group, which could be patient advocacy or families, to support the development. There are very clear, as I discussed earlier, sponsored activity for finding the right drug and doing the right studies, making sure it's safe. But on the patient-led activities, and these all have to go together to ensure a clear path and a fast path for approval for a rare disease therapy. And I would highlight the natural history studies and registries that are so important, and will by not only giving us understanding on the disease progression, but also biomarker, getting collection of biomarkers longitudinally, and finding clinically meaningful endpoints, which the FDA is interested in, because they want to see not what's important for the company, but what's important for the patient, and how you're improving the outcome. And also tissue biobanks are really important, and making sure that you have the right diagnosis. So all these activities have to go hand in hand with the sponsor, which could be an academic lab or industry or a foundation, to really integrate all this knowledge to have the best target product profile and have a fast track for finding more drugs and finding more therapies for rare diseases. I will stop here. I'll thank everybody who contributed to the two programs, Lafora, Ionis, and Cystic Fibrosis, Reductase, and Cystic Fibrosis Foundation. Thank you for your attention, and I look forward to your questions at the end of the session. And with that, I'll move forward and hand it to Ethan Perlstein.
Great, thanks, Tamar. Let me share my screen. Okay, today I want to tell you a story about personalized drug repurposing, where we were able to go from bench to bedside in about a year. And this is an example of building a plane in flight, but I think we've validated the blueprint, and now I think this blueprint can actually scale, and not just within this archipelago of ultra-rare diseases I'm going to talk about, but even beyond. So I want to tell you a story about a group of diseases called congenital disorders of glycosylation. There are 150 different genes and counting that, when mutated in an autosomal recessive way, so two mutant copies from each parent, they cause this multi-system progressive disease. And Maggie from Michigan has PMM2 CDG, which is one of these 150 ultra-rare CDGs, but it happens to be the biggest of them. And by big, I mean still only 1,000 patients globally. And most of these patients are kids like Maggie. And Maggie's family embarked on a cure odyssey with Perlara about 3.5 years ago. And today, Maggie is the first child taking a repurposed drug from Japan that we discovered together through a collaborative process. And the drug is working. It's not a cure, but it's working. And we're very hopeful that this N of 1 can be expanded to N of 1 plus. And the way we're able to move so rapidly and without sacrificing precision medicine, and even in this case where there was no mouse model available to us, we're able to put together a constellation of patient avatars consisting of yeast, worms, and patient-derived fibroblasts. And we were able to take advantage of the fact that congenital disorders of glycosylation are they involve very ancient pathways and very ancient genes. So you can model you can essentially model these diseases all the way in simple cells, simple single-celled animals like yeast. And here's just an example or here's a schematic of how this pipeline works, and I'll give you the real example in a second. So imagine a gene where you've got 3 different mutations, and they're color-coded here. And we know exactly what these mutations do to the protein. And that's not always the case, but we have that kind of deep biochemical insight in the case of a gene that's so ancient and well-studied like PMM2. So we know in the case of the red mutation that PMM2 can't form a dimer anymore, and that's essential for it to work, to be in this dyad. We have other flavor of mutation, this purple mutation, which causes the dyad to form, but then it falls apart, or one part of it is really unstable. And then we have...
Sorry, are you advancing your slides? Because we're not.
No.
Oh,
yeah, sorry about that. So yeah, just got to make sure the screen doesn't fall asleep. So, and the purple and the green mutation represents a case where you have a very pinpoint a kind of Achilles heel mutation where the dimer forms, it's stable, but the dimer simply has no output. It's imagine you pull the spark plug out of a car and it looks fine, but it just simply won't start. So we can model each of these mutations in yeast and in worms and flies, but principally and primarily in yeast, we can model them, and we can show that these mutations cause a growth defect that's kind of proportional to how severe they are in humans. And we can exploit that growth defect to do drug repurposing screens. And we can model all types of scenarios where we've taken advantage of the fact that even though yeast are super simple and all we can measure is that when they're sick, they don't grow well, and we have to look for a drug that can make them grow well, we know that they're not growing well for different reasons, as I explained, based on the mechanism of these different mutations and how they have, how they might respond to different therapies. So that's again how you don't sacrifice precision medicine, even though you're using a very simple model, which doesn't have all the organ systems and complexity of a human with PMM2. But we were able to find molecules in this yeast screen that acted to rescue growth in very interesting ways. And we weren't satisfied just sort of with understanding their growth patterns. We wanted to look at the structures of these molecules and understand where they come from to have an insight as to, are any of these clinically actionable? So one of the molecules in the top row, this alpha cyano4 hydroxymic acid, is actually a natural product that comes from cinnamon.
Sorry, we're actually only seeing your title slide.
I'm sorry, there's something weird
going on.
on.
10 people suggested you stop it and reshare it, and that might help.
Sorry about that.
Or are you not sharing from the, not advancing from the right screen?
Yeah, 1 second here.
Because it doesn't look like your screen is on.
No, because unfortunately, yeah, the PDF when it expands doesn't apparently screen.
Okay, might be really helpful for you to just show us quickly the slides we missed and catch up.
Yeah, apologies about that. So this is Maggie. This is sort of that schematic of yeast, worms, and fibroblasts. And how the different mutations are having they're breaking the function of the gene in a different way. And so we don't want to sacrifice precision medicine and trying to get therapies that might work for all or some or specific sets of these mutations. And so we were able to exploit, again, this very yeast-centric or very simple growth phenotype and just rescue that and then be able to use that as a starting point for testing the hits that we get in higher systems. So here are the structures of the compounds that we were excited about that were the most promising yeast hits out of a collection of about 2,600 repurposable drugs. And the first compound is an active ingredient of cinnamon. The second compound is a natural product, is sort of the active component of black tea. And the third compound was actually a very old FDA-approved drug, but it's for African river blindness, and we don't really understand its mechanism. But the first two compounds are these so-called polyphenols, and they've come up before in other screens, and some people have even said that they're too often hits, and so therefore they're not really specific. But this got us thinking, well, what if we did the same set of screens, but this time in a worm model? Because the worm's more complex, more cells, it's multicellular, doesn't have organ systems per se, but it has neurons and other differentiated cell types. So we recreated this one flavor of mutation, the so-called F119L mutation, which is the second most common mutation seen in patients. And this is the mutation, again, that causes the dimers to no longer form. And we could show that by measuring the output of the dimers, the output of the PMM2 enzyme, we could see that in orange, the mutant that we generated has lower levels of enzyme activity. So it's a bona fide PMM2 worm avatar. And we could do simple, again, simple from the point of view of we're just looking at the way these animals are growing and reproducing in a confined environment like a tiny well. And so we can see here that the principle of a phenotypic screen where you say, I've got these PMM2 worms, they're sensitive to a particular drug called bortezomib. And so you see that they're not really growing well. There's not many of them. And then we can set ourselves up to then look for a molecule or suppressor that can make these PMM2 worms that are stressed grow better again. And of course, we could find molecules that make them worse, but we're really excited about suppressors. And we really wanted to see whether if we did the same screen or if we did the worm screen on the same collection of compounds that we screened the yeast on, what would that convergence look like? And what we found was that there is a class of drugs, very old class of drugs actually, called Aldose reductase inhibitors. And what we found was that cinnamon compound and some of the other natural products that are of this polyphenol class, they actually resemble a known drug on the market, but not in the US market and the Japanese market as of 1992 actually. And what you see here is in orange and blue the components of the molecule that are shared, and you can kind of see why these molecules are related. So I'm just showing you a doublet of the cinnamon compound just so I can highlight the cinnamil region, which is obviously you can see where the name cinnamon comes from. And then you can see this other region in blue, the carboxylic acid, right? This is the same molecule. I'm just separating them so you can show the orange bit and the blue bit, and you can see that they're basically fused together in this this drug that is called the Powerstat, or Kinodac is the brand name. And we were very excited to ask, well, if this is an approved drug, is it acting by the same mechanism as this natural product? And we could do that experiment by looking side by side at the PMIM2 enzyme activity in either worms or in patient fibroblasts, and see that our drug, a Powerstat, was boosting enzyme activity over the baseline. Again, it wasn't a complete rescue in this particular fibroblast or genetic background, but both the cinnamon compound and the Powerstat compound were boosting PMIM2 activity in worms and in cells. So the yeast acted as sort of this springboard, and then we were able to identify an actual mechanism of action in a known drug that we think is specifically acting on the PMIM2 pathway, and that's never been shown before. There's never been a drug that increases the level of PMIM2 activity. So that was really exciting to see. We tested multiple different fibroblasts with our clinical collaborator, trying to understand if you took different mutations of PMIM2 and asked, are they all, could they all benefit from this Powerstat? And it looks like by and large, the three that we tested all showed an improvement, some more than others. Some showed a 400% boost over baseline, some, like the slide before, one cell line only had a 40%, but it was a pretty big range, and on average about twofold increase in PMM phosphomonomutase activities. That's, again, no known drug, novel or otherwise, has been reported to directly boost the activity of this enzyme. So that was really gratifying to see. And then we wanted to take all this theory and translate it. And so this is where we went into kind of overdrive and said, let's go from these discoveries with yeast and worms and fibroblasts and these biochemical insights that made sense, and say, do they check out in a human? And working with the family, the ProQuest family that started to fund this work years ago, we were able to rapidly go through these FDA sort of hurdles. And first was getting a single patient or compassionate use IND, which allowed for this N of 1 study to begin. So we talk about N of 1 colloquially, but FDA uses the language single patient IND, or also compassionate use IND. We were able to then get an orphan drug designation, which allows us, so in the scenario where we market this drug, we're able to get certain benefits. And then the rare pediatric disease designation as well is a benefit you get, where if you get a drug approved for a rare pediatric disease, you're able to get what's called the priority review voucher, which has value on the biotech market. So we were able to go through, I can go in detail over these processes online or offline, but we were able to go jump through these hoops and do first patient, first dose earlier this year with Maggie, her doctor, her mom, and the clinical trial coordinator. And we're able to show that so far the drug is well tolerated. So this drug had been given to adults for diabetic neuropathy for years in Asia, but never tested in a child with this kind of disease. So that was question one: is this well tolerated? And thankfully, it looks like there have been no serious adverse events, and the drug is well tolerated. There have been improvements in speech and stability that the family and caretakers and others have noticed over the last few months. There's been normalization of blood, and what we're most excited about is a urine biomarker. And I'm just showing you here without identifying the markers yet, because we're in the process of expanding this data set and publishing it as soon as possible. But excitingly, this is a log scale. So before a Powerstat treatment, this marker was elevated to a 648 count, and it's now 37.32. So we hope that this biomarker can be further validated, because it's very easy to get urine and very easy, therefore, to do a trial in spite of COVID, where you could do this in a distributed way and still get a lot of meaningful data and show target engagement by your drug. So the 6-month visit is approaching for this N of 1. We're getting ready to schedule a pre-IND meeting with FDA and later in the summer to get clearance to expand the trial to a larger set of patients and to try higher doses as well. And this is, again, not just a one-off. We're already showing that with a sort of sister disease, NGLY1, another glycosylation disorder, we're gearing up to do the same thing. Matt Might, a good friend, mentioned this in his session earlier today. I won't have much time to go into the science of this, but suffice to say that you heard Buddy's story about being patient one with NGLY1 deficiency. And what we found, using a very similar process, except with a different trio of models here, we used flies and worms and cells, and instead of yeast, we use flies. But we were able to show that aripiprazole, which is the generic name of Abilify, which is a known psychoactive drug, actually approved for use in children as young as 9 for attention deficit disorder and for some certain psychiatric conditions. We were able to show that aripiprazole is the top hit for NGLY1 deficient models, both rescuing the fly and the worm, but also in boosting this pathway called NRF2 pathway, which is the pathway that Matt Might mentioned where his AI was trained on the literature and said, hey, you've got to boost the activity of this gene called NRF2. And lo and behold, this aripiprazole, which in an unbiased way rescues flies and worms that are NGLY1 deficient, it also boosts the activity of this critical disease-modifying pathway. So we're very excited to be moving into a clinical trial here as well, with Buddy being N of 1. And at the center of this is a really great collaborator and great human being, Dr. Eva Morava, who is kind of PI for both the NGLY1 trial and for this PMM2 N of 1 as we seek to expand it. And we couldn't have done this without the families. It's not a traditional, Prolara is not a traditional biotech company. I'm not showing you the board of directors. I'm not showing you all the stodgy old white guys on the SAB. I'm showing you the actual families that made this possible, who are the de facto co-founders and therefore de facto the proper co-owners of the work that we're doing. So we're excited to actually be spinning out a new co with Holly, Maggie's mother, and their family, so we can develop the Palrostat, hopefully in the US of A, if this is approved, for pediatric formulation and kids with CDG. So we're excited to move forward with CDG, but we think that this model could work for other diseases of metabolism and more broadly. So happy to take questions and pause here. And I really appreciate Tamar setting up this talk and giving you the bigger picture when I zoomed in here for this particular N of 1. So thank you.
Thank you, Tamar and Ethan. We have some really interesting detailed questions here coming in. So the first one is for Tamar. Can the lunar technology be applied to brain-based diseases? Do the particles cross the blood-brain barrier?
Thank you for the question. This is an excellent one. This is exactly the question that I asked when I joined Arcturus, is can we get to the brain? Can we treat neurological diseases? And the answer is not yet. We are working on it. There are lunar formulations could target different cell types, and we are working currently on trying to target neurons and other cells in the brain to open up opportunities for us to treat the neurological diseases. And to answer the second question, lunar formulation do not cross the blood-brain barrier. It depends on what formulation we use, but most of the ones that are in the clinic or that are going into clinic will be those treating liver diseases, and the next one will be the one that I described today to target the epithelial cells in the lung.
Okay. Thank you. This is also a follow-up question. The approach Tamar has presented can be also used, this is a question, can it also be used for those CF patients having missense pathogenic mutations?
Absolutely, and I'm sorry if I didn't emphasize that. Thank you for the question. The lunar CFTR drug can treat any mutation on any for any of those CFTR mutation, whether it's just generating, and there are different classes of mutations for cystic fibrosis. Some of them will generate truncated protein, some of them will not generate a protein, some of them will have a protein that will not translocate to the right compartment of the cells, and that will cause fibrosis. But for all of those, these are all dysfunctional or non-existent proteins, and the lunar technology, the messenger RNA, will basically introduce the normal, healthy CFTR protein and will be potentially beneficial for all mutations.
Okay, great. Let's see, one of the questions has, where did it go? Ethan, are you opening NUCO in order to apply for government funding, SBIR and SSTR?
Yeah, and any kind of funding to make it fundable. And that was always the Perlara model, was to be able to create subsidiaries that are built around clinical focused assets and do that where that NUCO is co-owned by Perlara and the family that funded the work to get there. That was always the point.
Okay. And I see you answered one of these questions, but I'll read it so the whole So the whole crew gets to hear it. I was under the impression that doing a high throughput drug repurposing screen in yeast was less feasible than in mammalian cell models, fibroblasts, because yeast cell wall inhibits application of compounds. How did you get around this?
Yeah, I answered that by saying that's just a decades-old mythology, and it's just a non-issue. It's just simply not true. The cell wall does require you to use higher doses that you might, at the primary screen level, that you might do in cells, so 25 micromolar instead of 10 micromolar, but it's not like you have to do it at 100 micromolar or higher. And the reality is it's just a non-issue. I've been dealing with that issue for decades. It's a non-issue.
Okay, so we had one more opportunity to dispel the myth today. Speaking of translational research moving from academia to industry, what are some of the key best practices for advocates who want to work with industry that you would recommend?
So I think it depends on kind of where the knowledge base is for your community. If you're at a place where you don't know enough to have accredited targets, and you don't know what mechanism to pursue for disease modification, then you might have to sort of go you might have to go a little slower. But if you've got a validated target, then it's possible to, I think, work with small biotechs for sure. But also, I think you could imagine pursuing an SBIR or STTR pathway. Sean Eakins from Collaboration Pharma talked about that and their model, which is kind of purely grant funded. So it's slower, admittedly, but if you experience with that system, it's a pool of funding. Tamar, you have any thoughts?
Well, I think it's really key for the patient advocacy group who really wants to push forward and get the drugs into the clinic to communicate and interact with the industry at all levels. I mean, a lot of my rare disease programs were initiated in conferences, and where I gave a talk and talked about the capabilities and the technology, and then patients, families in the audience will say, well, can you do something for my child? He has so-and-so disease. So just not, don't be shy, and just try to make the connections and the interactions. And I think regarding activities, I think if those patients organization can embark on, and we've heard about it today from Nasha, that was really fantastic overview of what activities and what's critical for these foundations and patient organization to do to really support pharma, will be the natural history studies, because these are really key to move drugs forward and to initiate them either in partnership with pharma and the natural history study that we're running for Lafora disease is done by two companies that are interested in taking the drug forward to the patient, and there's not that many patients out there, so there's no point of running multiple natural history studies. It has to be a combined collaborative effort between the patient groups, but also industry and multiple companies that could also work together to make things happen and just get the information needed to push those drugs forward, and have those trials that are without the placebo control, making sure that all patients are getting dosed. And all the other activities that I also mentioned on the biobanking and registry, we're talking about ultra rare diseases where there are multiple, patients that are scattered worldwide, and just getting together. And there are all these Facebook groups and all kinds of social media activities that are happening, and trying to group everybody together worldwide and try to form those registries and group the patients together. I think these are really critical activities that I would highly encourage organizations to follow.
Okay, great. So do you have any advice in terms of making sure that those registries or natural history studies have shared data, and that it's accessible to the most number of researchers who have relevant research?
Yeah, this is a tough one. I mean, it really depends. I think this needs to be kind of communicated and worked up from the get-go when you start working on these registries, and try to keep those. And usually with academia, things are done very with transparency and publications that are out there. So I think working with the academic groups and industry and making sure that the information will be available for for everybody. That's something that needs to be worked from the start, because I know that some pharmaceutical companies will embark on those studies and just keep the data for themselves, and this is just not a good practice for the community.
And how do you know how much basic research has to be done in your space in order to develop the knowledge base that will support translational research? When do you know enough is enough, I guess?
That's a very good question, and it really depends on the disease and the disease models. What we are trying to aim for with the rare disease, and that with my experience, is to have a proof of concept in a perfect disease model. It's usually a mouse model where you show that the drug is improving disease. And but there are many programs, and the cystic fibrosis could be a good example for that, where there are no really good models that recapitulate the whole pathology of the disease. There's some that are showing, you know, like the mouse model that have the knockout, but doesn't have its other symptoms. It just doesn't have the channels. There's the rat model that generate the mucus, but not the other. So for example, for the Vertex drug, all the proof of concept data that was required to take the program forward was relying on data that was generated in human cells. So when you don't have the animal models, you can go, and there's like precedents in the FDA of taking drugs forward with showing proof of concept in the cells. But I think proof of concept data is critical, whether you do it in cell lines or whether you do it in the animal model. I think that gives the rationale, and also for the efficacy, but also for safety, to know that when you target it in the animal models, you have the safety. But you can always generate more and more and more data. So it's just it really depends on the program and the sponsor who would want to take program forward to know where to start and when is enough to take that forward to regulatory. Ethan, do you have anything to add?
No, sounds good.
Thank you.
Then I will ask you both one last question. What excites you about rare disease research today?
I think what excites me most is, again, Matt Might made this point earlier, you know, if you were watching N of 1 5 years ago, it was really only accessible to either the wealthiest or most connected families who happened to hook up with the most kind of all-star elite researchers who were willing to take these risks of pursuing experimental therapy. It's no longer the frontier. Now there's a lot of examples, many N of 1s you could point to. So now I think we're entering an era of consolidation and scale, where the priority is accessibility to these N of 1 approaches. So there's no longer financial resource and sort of social graph barriers, so that anybody who presents with a mutation and you're kind of blind to everything else, you would just sort of match with the appropriate modality or combination of modalities. So I'm just excited by this decade of what I think will be cures, because if you look at the SMA example, spinal muscular atrophy, you have to say to yourself, is that a fluke? Is it a fluke that one disease is now benefiting from a gene therapy, an ASO, and a small molecule, right? Is that just going to be the only case that happens? I mean, clearly not with CF. CF have a triple combo with that's pharmacological. Now we see drugs like what Tamar is talking about that are going to take care of the rest of the population, disease population. So I'm really encouraged by just how curative this decade will be. And you can quibble about what's a cure and whether it should be singular or plural, but I'm just excited by the fact that we're no longer just experimental phase with N of 1, we're in scaling phase.
Mm-hmm. Right. And Tamar?
I couldn't agree more. I mean, I'm super excited about all these initiatives and the N of 1 and the successes that have been showing in the clinic now. And everything that Ethan's doing, I think that's the future, this of precision medicine. There are all kinds of initiatives that are nonprofit that are going after those ultra rare and N of 1 diseases. And when I started working on rare diseases 10 years ago, I've got a lot of no's for programs that I tried to initiate when they said, well, there's not enough patients. How are we going to do a clinical trial? But now the past couple of years, we can see that we can have clinical trials done, and we can have a handful of patients showing efficacy and get a drug move forward and talk to regulatory and agencies and have it approved. So I think this is a very exciting time. And in the next decade, like Ethan said, we're going to see more and more rare disease drugs that are coming to the clinic. And I think this is also due to very exciting advancement of technologies, especially in the RNA space, where we can not only knock down genes that are problematic or going to have benefits, but we can also upregulate genes. We can also use TALEN. We can use all kinds of approaches like the messenger RNA and basically correct all diseases that have mutations. And it's just a matter of enhancing our technology where we can really target CNS, and we can target muscle, and we can target all kinds of different diseases. So these technologies and the changes in regulatory and those new foundations and new companies that are coming up to really focus on rare diseases is going to really pave the path for really great drugs that are going to be out there and helping save patients with the disease.
Thank you so much, both of you. And we do have some more questions in the chat, so we'll follow up with you if you don't mind, and we'll get those
Absolutely
public. And I don't want anyone to miss the closing session, so I will just say very deep gratitude. Thank you to both Ethan and to Tamar for coming and sharing all your knowledge today. And thank you all for attending.
Thank you.
Welcome. Thank you.